Hello, everyone. My name is Katarina Berk, and over the past 8 weeks, I worked in the Hofstetter lab. I worked on the directionality of secondary injury after traumatic spinal cord injury, the histological validation of contrast-enhanced ultrasound. Traumatic spinal cord injuries, or tSCIs, often result in permanent sensory and motor damage, leading to debilitating effects like paralysis and incontinence. And the main causes for these injuries include falls and motor vehicle collisions. And the US ranks among the countries with the highest incidence, as can be seen on the right. In addition, 17,000 new traumatic spinal cord injury cases occur annually in the US. It's important to note that there are two phases to the injury. There's the primary injury and the secondary injury. The primary injury is that initial contact with the spinal cord, and the secondary injury is how it expands over time through multiple processes such as apoptosis, inflammation, ischemia, and many more. If you look to this primary MRI here, you can see the primary injury. And if you look to the 72 hours post-injury image, you can see the difference in the arrows, and this shows the secondary injury expansion over time. It's important to note that the primary injury is essentially irreversible, but the secondary injury has room for possible treatment to intervene, which is what we're most interested in. And recent advances in contrast-enhanced ultrasound, or CEUS, have allowed us to visualize the vasculature of the spinal cord and enabled these rostral-caudal injury expansion studies, rostral being towards the head, caudal being towards the tail. For our methods, we use two cohorts of Long Evans rats, one to image the cervical spine and one to image the thoracic spine. A laminectomy was then performed, and this removes the outer protective bone, exposing the spinal cord and creating that acoustic window for the ultrasound. The moderate bilateral contusion was then performed to act as the traumatic spinal cord injury, and CEUS imaging was done throughout the procedure. The baseline was taken prior to the injury, acute was done directly after the injury, and the final imaging was 72 hours post-injury. If you look to the right, you can see a visual of what our setup looked like, and this bolus injection of Definity is the contrast agent we injected to allow us to visualize the vasculature on the ultrasound. We did notice some directionality in these images. So if you look at the two acute images for both cervical and thoracic, the acute injuries look relatively the same, but when we jump to that 72 hours post-injury mark, you can see that the cervical has a more symmetric expansion compared to the thoracic, which has an exclusive caudal expansion. And to validate our results, I did the histology portion this summer. I sectioned all the rodent spinal cords at 20 microns thick and around 1.5 centimeters in length. I then performed staining. I did immunohistochemistry with the target protein caspase-8. Caspase-8 stands for apoptotic cells, or in other words, programmed cell death. I also stained for crescent violet and areochrome. Crescent violet stains for the Nissl bodies and the neurons, and areochrome stains for the myelin sheath. I imaged these using the ActioZoom microscope for the immunofluorescent slides, the caspase-8 slides, which can be seen on the top image here. So those fluorescent points are the apoptotic cells near the lesion center, and the Nikon Eclipse microscope for the crescent violet and areochrome slides, which is the bottom image there. And you can see clearly the lesion cavity. As for quantifying the results, I was blinded for the quantification, so I didn't know if I was looking at cervical or thoracic tissue. And I analyzed these over 7 regions of interest located near the epicenter. We expected to see results that were similar to those seen on the ultrasound. However, that wasn't quite the case. Based off this graph, thoracic showed a more symmetric expansion, and cervical had a relatively symmetric expansion with a slight caudal shift. And there could be many reasons why these are the results that we saw. Histology does have limitations, and the histology and ultrasound comparison were done over different temporal ranges. So ultrasound was done over the 72 hours, and histology is just at the 72 hours mark. So there is that limitation, as well as I only stained for one marker. There are several markers for cell death, and I also analyzed a limited region. So there's plenty of room for re-quantification of this data by adding additional stains or possibly expanding that analysis of the cord. As for clinical implications, the ultimate goal would be to translate this from the rodent model to the human model, looking at that comparable directionality between the two. And this could see applications like taking a closer look at those post-surgical MRIs or CEUS, which is this image here, after traumatic spinal cord injuries in humans, and using them kind of as a diagnostic tool to be able to identify the salvageable tissue after those injuries. Here are my references. And I'd like to give a big thanks to everyone in the Hofstetter Kind Lab, my PI, Dr. Hofstetter, my lab mentors, Yannick Lanebecker, and a special thanks to Jen Harmon and Z. Cates, who worked on the ultrasound portion of this project. And I'd also like to thank Dr. and Mrs. Ellenbogen, Julie Bold, Sylvia Savachkin, and everyone else who made the program happen. Thank you.